Hi everyone. I thought we would talk a little bit about blood vessels, particularly the structure of blood vessels and then the characteristics of the different types of blood vessels that are found in the human body. So we're going to just start off by drawing a sort of generic blood vessel with all the possible structures, and then we'll eventually break down which blood vessels have which components. So what we're drawing first here is called the tunica intima, and it's actually the innermost layer of the blood vessel. And so we can see, like, right in the middle, we have the lumen of the blood vessel, so where the blood is contained. And the layer of cells that basically surrounds the lumen, or the innermost layer, is called the endothelium. And it's basically simple squamous epithelium. The simple squamous epithelium is not, it's continuous in most cases, but sometimes there are some gaps called intercellular clefts. And that's a possibility for certain things to sort of squeeze between the blood vessels to move out of the blood compartment. Because it's epithelium, of course, it's surrounded by a basal lamina, and then we have what's called the subendothelial layer in most cases, except for maybe the capillaries. And it's made of loose connective tissue and elastin. The elastin is typically only present in arteries and arterioles, and it's actually labeled the internal elastic membrane. The second layer is the tunica media, and so it's basically concentric around the inner layers. And the tunica media is basically layers of smooth muscle cells and then layers of elastin. And so basically these layers are also concentric around each other. So the smooth muscle cells are surrounding the internal elastic membrane, and then we have a layer of elastin, and then we have more smooth muscle cells, and then more elastin. And the elastin is actually fenestrated, meaning it has some holes or pores. It's usually similar to, so we actually drew them with the subendothelial layer as well. And that actually allows for diffusion between the elastin to the different cell layers. And then the final layer is the tunica adventitia. It's also called in some textbooks the tunica externa. And this is kind of a thick layer, and depending on depending on which vessels we're talking about, it may or may not be present. And basically it's bundles of collagen fibers. And so the collagen is actually what gives something like a blood vessel some sort of strength and support and ability to recoil after stretching. And it also we have some scattered elastin, and we have certain types of cells, particularly fibroblasts and macrophages. Fibroblasts are probably the most prevalent form of cell for connective tissue, and macrophages engulf bacteria, cell debris, things like that. We have nerves that run through this layer, and then we also have what's called the vasa vasorum, which is basically blood vessels that supply the blood vessels, which is kind of crazy to think about, but because the tissue layers are so thick and we're talking about cells that need to survive, we need to have good perfusion, and we can't have exchange of oxygen and nutrients all the way from the blood to this outermost layer, so we have blood vessels that are very small that supply that particular area as well. All right, so let's sort of just trace our way through the different types of vessels, and we'll work from arteries to capillaries to veins. So we know that blood leaving the heart leaves through arteries, and there are sort of two major types of arteries. The first one is elastic. Elastic arteries are about 10 to 25 millimeters in diameter, and they contain elastin between the smooth muscle layers, and the smooth muscle cells in elastic arteries are not active in vasoconstriction. So the purpose of elastic arteries is to stretch to accommodate blood entry, and then to recoil, and stretch, recoil, and not necessarily constrict to limit the blood flow. Now, the common examples of elastic arteries will be the aorta, the pulmonary trunk, pulmonary arteries, and then some other ones that I don't think is maybe necessary to memorize, but you could certainly view it in a book. Common carotid, the common iliac, and subclavian arteries. The other major type of arteries are muscular arteries. These are a little smaller, more like 2 to 10 millimeters in diameter. And these arteries basically deliver blood to specific body organs. So we start to break down, instead of delivering to like a body area, like the upper or lower portion of the body, we talk about which organs in particular the blood is going to. So in comparison to elastic arteries, they tend to have more smooth muscle cells and less elastin. And they're actually more active in vasoconstriction, and so therefore they're less capable of stretch. So a little bit of the opposite in comparison to the elastic artery function. Some examples of muscular arteries include the external carotid arteries, the brachial arteries, the femoral arteries, and the mesenteric arteries. And then the last type of arteries, sort of artery, I guess, that we want to talk about is arterioles. They're like little mini arteries. And they can be about 0.01 to 0.1 millimeters in diameter. So now we're getting very, very, very small. And these have pretty much endothelium and smooth muscle cells, but they don't have a tunica adventitia. So they pretty much stop at the smooth muscle cell layer. And these guys are able to change their diameter in response to local neural and hormonal stimuli, which is something we'll cover in the next video. But these guys are responsive to sympathetic activation, to parasympathetic activation, to different types of hormones, that sort of thing. Next, let's talk a little bit about capillaries. So we've mentioned before that capillaries are responsible for exchange, are the sites of exchange of nutrients and oxygen and carbon dioxide at the tissue level. But we need to sort of expand and add some details about the capillaries so that we're all so that we have a more thorough appreciation of what they do. So capillaries are about 0.004 to 0.01 millimeters in diameter, so teeny, teeny, tiny. And they are basically endothelium plus basal lamina. And so they don't have a tunica media or a tunica adventitia, which is good because we want to be able to have good diffusion. So we have these cells called pericytes that sort of hang out and sort of surround, not necessarily entirely, it's not a full enclosure, but hang out around the endothelial cells, and they function to stabilize the endothelium and control capillary permeability. Of note, these cells are stem cells, which basically means that they can become anything. And so if there's an injury to either endothelial cells or vascular smooth muscle cells, these guys can divide and differentiate into either one of those two cell types to sort of replace the dead tissue. There are three types of capillaries. So the first type of capillary that we want to consider is the continuous capillary, which basically means that most cells are joined by tight junctions, and there are few intercellular clefts or gaps between the cells that offer the opportunity for movement of different materials into and out of the capillaries if they can't pass through the actual cells. The second type is fenestrated capillaries. And fenestrated means that there are pores in the endothelial cells that basically increase permeability. And so these guys are still covered by a basal lamina. Even the pores are covered by a basal lamina. It's actually called the diaphragm. And it doesn't prevent things from moving out of the pores. It just offers a little bit of control over what actually is able to move. So commonly we'll find fenestrated capillaries in the digestive tract, in the kidneys, and certain endocrine organs, and in the choroid plexus, which is where the CSF is made in the brain, in the ventricles. And the last type of capillary is the sinusoidal capillary. So sinusoidal capillaries have, these are considered leaky capillaries. They actually have large gaps in between the cells, and the gaps are so big that some blood cells can even pass through. So these will commonly be found in the liver. So like in the liver, we'll have macrophages that hang out right outside the endothelium, so they can engulf bad stuff that we don't want to move anywhere else in the body. We also will find them in the spleen. Similarly, we have phagocytes. So same kind of idea. Phagocytes, macrophages, both engulfing stuff. And the sinusoidal capillaries are still covered by basal lamina, but the basal lamina is more discontinuous than covering the entire structure. So let's take a look at the capillary network and sort of understand some of the components that allow for exchange in the tissues. So we have our arteriole, and the arteriole will sort of branch into a meta-arteriole. So that's kind of this first part across the way here, and then the second part sort of becomes more like a vein, capillary vein, is called the thoroughfare channel, and that will bring us back to the venule. So together, these two items, the meta-arteriole and the thoroughfare channel, are referred to as the vascular shunt. So the blood will flow in the arteriole and it will flow across the vascular shunt into the venule. And then certain factors will determine whether or not that blood also enters into the true capillaries. So the purple is the true capillaries, and then we can actually see that we have these little precapillary sphincters that surround the entryways into the true capillaries, and those are smooth muscle cells that make up the sphincters. And so basically, if the smooth muscle cells are relaxed, then the blood flows into the meta-arteriole and also flows into the true capillaries, and then we can have exchange of the peripheral tissues. Conversely, if the smooth muscle cells are contracted, that means that they're sort of sealed off and the blood can't pass into the true capillaries, and it just passes through the meta-arteriole to the thoroughfare channel and directly to the venule. So in cases where we need more blood flow for the body, if we are trying to work on temperature regulation or if we're hypovolemic, then we'll just sort of shunt the blood through the meta-arteriole and the thoroughfare channel. And then when that's not the case, we can have exchange, we'll actually use those true capillaries. And just of note, the vascular shunt and the true capillaries together make up what's called microcirculation, or the capillary bed. So in cases where we can have exchange, let's just talk a little bit about, and this might be a review as well, but let's talk about transport across the endothelium. So option 1 is that we could have diffusion, which is transcellular, so across the cell. And we didn't label our epithelial cells, but we could just label the lumen and the basal lamina so we can keep track of the direction. The second option is we could have transcytosis, so endocytosis and exocytosis. Third option is paracellular, meaning transport between the cells. And then fourth option is that we're in a case where we have a fenestrated capillary, we can pass materials through the fenestration, through the pores. So that leaves us lastly to talk about veins. And so we'll start with venules, so the little teeny tiny veins. And these guys are approximately about 0.01 to 0.1 millimeters in diameter. And so we can see that we're starting to go back up a little bit, get a little bit bigger. Venules, there are different sizes of venules. And so the smaller ones may be endothelium only, and they tend to be kind of porous or leaky. And the bigger ones will be more likely to have 1 or 2 layers of smooth muscle cells that are kind of scattered, and they may have also a tunica adventitia. Now, in terms of veins, we tend to classify them as small, medium, and large. And so we'll just kind of talk about the different diameters of the different sizes. So small veins would be like 0.1 to 1 millimeter in diameter. Medium would be like 1 to 10 millimeters in diameter, and large would be about greater than 10 millimeters in diameter. A large vein would be something like the superior or the inferior vena cava that directs the blood flow into the heart. So in comparison to arteries, veins tend to have thinner walls and larger lumens than arteries. So a lot of times if we're looking at, like, for example, a slide of them, a cross-section of a vein and an artery, we would actually see that a vein tends to be a little flatter, doesn't retain its shape as much, and an artery tends to be kind of rigid in structure. The veins usually have very little smooth muscle cells, and they also carry blood at very low pressure. So when we think about getting to the end of a capillary, we're probably looking at about 18 millimeters of mercury, plus or minus. So basically, the trip from venules back to the inferior vena cava and the superior vena cava into the heart is a decline from about 18 or so millimeters of mercury to about 2 to 6 millimeters of mercury as we enter the right atrium. So it's a very low pressure, which means that there's very little resistance to blood flow. So the blood can flow very easily. But the downside is that if it's very low pressure, sometimes, for example, the blood has to fight gravity to make it back to the heart. So blood that's coming from the lower limbs has to travel upward. And things like skeletal muscle contraction on a kind of like walking can help to improve blood return. But also the veins will contain valves. And the valves are basically folds in the tunica intima. And the valves, so they sort of just sit very similar to the valves in the heart. So they sit nice and closed, and then the blood flows up through it, opening the valve, and then any backflow, the valve will close on itself and prevent that blood from traveling beyond the valve in the backwards direction. So that is all for now. Hope you enjoyed it. Thanks for watching.